Welcome to the Deep Dive. Today we're really getting into the engine room of evolution itself, speciation. How new life forms, that exact moment one species sort of splits into two. It fundamentally changes everything. We're using a pretty rigorous chapter from Strickberger's Evolution as our guide here. And our mission really is to pull out the core ideas, the essential concepts, the mechanisms, and maybe some surprising evidence, especially focusing on that one absolute requirement for speciation.
Right, and that requirement is reproductive isolation. It's absolutely central. The biological species concept really hinges on this. We have to stress just looking different isn't the key. A species is defined by its inability to interbreed successfully with another group and produce fertile offspring. That barrier, that's the crucial thing.
That makes sense. The reproductive barrier is non-negotiable.
Exactly. And we'll look at the main ways this happens. There's sympatric speciation, which is less common, happens within a population. But mostly we'll focus on allopatric speciation. That's where geographical separation kicks things off. It's overwhelmingly the usual starting point.
Okay, let's dive into that then. Allopatry. So it sounds like geography is kind of the main thing driving this split, right? Once populations are physically separated, what are the essential steps for them to actually diverge into new species?
Yeah, it's a sequence. Really crucial. 3 steps. First, you absolutely need that geographical split isolation. Second, the separated groups start adapting locally. Divergent natural selection pushes them in different directions based on their environment. And third, eventually those genetic differences pile up and create reproductive isolation. That's the final lock. It stops them from interbreeding, even if they somehow meet again later.
That separation is critical because it cuts off gene flow, right? The mixing that would normally keep them similar.
Precisely. It stops that homogenization.
And you mentioned 2 ways the split can start, like a big geographical event or just a few individuals heading off somewhere new.
Exactly. That second one, the few individuals, leads us straight to the founder effect. And the history here is pretty fascinating. It goes back to the 19th century. Reverend John Gulick, studying at Catanella land snails in Hawaii, Oahu specifically.
Snails!
Yeah. He noticed their distribution was incredibly localized, often confined to just a single valley, or sometimes even just part of a valley.
You can almost picture him, right? Walking these little valleys, mapping out snail locations, and basically stumbling onto a huge evolutionary idea.
That's exactly it. Gulick figured out that when a tiny group starts a new population, they only carry a small, possibly non-representative fraction of the original gene pool. This genetic bottleneck means they start from a unique genetic place, which can lead to really rapid, distinct evolution.
And someone else actually coined the term later.
Yes, Ernst Mayr. He called it the founder effect in 1952, building on Gulick's observations. Really powerful concept.
And the consequences can be huge, right? Leading to things like adaptive radiation. Darwin's finches in the Galapagos must be the prime example. That one ancestral finch arrives, and suddenly you get this explosion of different species filling all sorts of niches across the islands.
It's the textbook case.
Yeah.
Absolutely. And think about Australia with the marsupials. Because Australia was isolated, cut off from competition with placental mammals for so long, marsupials just radiated out. They filled ecological roles, similar to wolves, moles, you name it.
A whole parallel evolutionary track.
Exactly. It shows what happens when founders hit an open field, evolutionarily speaking. No pressure, lots of opportunity. Now, it's worth noting not all geographical separation works the same way. The chapter breaks down 3 modes based on how much isolation there is and if gene flow can still happen.
Okay, what are the 3?
First is allopatric speciation. This is the clean break. A physical barrier splits the population completely. No gene flow, period.
Got it. Total separation.
Second is peripatric speciation. This is basically the founder effect we just talked about. A small group buds off, isolated. Again, 0 gene flow, back to the parent population.
So allopatric and peripatric are both about complete isolation, just different scales maybe.
Yeah.
One splitting a large population, the other a small group budding off.
Pretty much, yeah. Clean breaks.
Okay, what's the third one then? How can species diverge if they aren't fully cut off?
That's peripatric speciation. Here, populations are adapting differently, usually at the edge of the species range, but they stay next to each other. They remain contiguous.
So like neighbors adapting to slightly different local conditions along a border.
Perfect analogy. They're diverging, but because they're still in contact at the edges, there's still some potential for gene flow, a bit of leakage across that border, even as they become distinct.
Interesting. So different degrees of separation, but ultimately it all comes down to actually stopping interbreeding, right? Those biological roadblocks you mentioned, the rims.
Exactly. Reproductive isolating mechanisms. It doesn't matter how the separation started, the process isn't truly finished until these rims are in place.
This is where it gets really specific. What kinds of rims are there? How do they actually work?
We generally classify them based on when they act in the reproductive cycle.
Hmm.
First, you have prezygotic mechanisms. These act before zygote can even form. They prevent mating or fertilization itself.
Like what?
Well, things like seasonal isolation. Maybe two types of spiderworts, Chaetoscantia, live in the same area but bloom at different times of the year, or prefer slightly different soil moisture. They never get the chance to cross-pollinate.
So timing is everything.
Or behavioral isolation. This is huge. Think species-specific courtship dances, bird songs, frog calls. Females only respond to the right signals from their own species. Plants might rely on specific pollinators attracted to unique floral displays.
Evolutionary gatekeeping, basically. Making sure only the right key fits the lock.
You got it. Then second, you have postzygotic mechanisms. These kick in after mating and fertilization have already happened. The hybrid zygote forms, but then problems.
Okay, so what kind of problems?
Could be failure of fertilization at a finer level. Maybe the pollen tube just can't grow properly down the style of the other species. Or the gametes might die, or the early embryo fails to develop. We see this in crosses between different rata frog species. The hybrids often die at predictable developmental stages.
Ah, developmental incompatibility.
Right, and probably the most famous example is hybrid sterility. Think of the mule.
Horse and donkey.
Exactly.
What?
A horse has 64 chromosomes, a donkey has 62. The mule offspring gets 63. That odd number messes up meiosis, the process of making sperm or eggs. So the mule itself is healthy but sterile. It can't reproduce.
That makes the Hawaiian Drosophila example even clearer. All those 800-plus species must have developed combinations of these rims after those founder events.
It's one of the most spectacular examples of speciation driven by founder events, absolutely. The pattern of colonization follows the age of the islands perfectly. Kauai is the oldest, about 5.6 million years old, and it has the oldest, most ancestral Drosophila lineages. The youngest island, Hawaii, was colonized entirely by founders flying over from the older islands. Each new island was a blank slate for a new founder event, a new burst of speciation.
So isolation gets the ball rolling. But is there anything that actively speeds up the divergence once populations are separated? This brings us to Alfred Wallace's idea about hybrid inferiority, right? Back in 1889.
Yes, Wallace had this key insight. His logic was, look, if two populations have adapted to different environments, any hybrids they produce are likely to be, well, less fit, sort of stuck in the middle, not well suited to either parental environment. If those hybrids are adaptively inferior, then natural selection should strongly favor any trait in the parent populations that prevents hybridization in the first place, things that strengthen sexual isolation.
Because making unfit hybrids is an evolutionary waste of time and energy.
Exactly. Selection will favor individuals who are choosier about their mates, reinforcing the species boundary to preserve those well-adapted gene combinations.
And there's data to back this up from Drosophila again.
Oh yeah, really compelling data. They compared pairs of closely related Drosophila species. When species pairs live in the same area, they're sympatric, meaning their ranges overlap and they encounter each other. Their level of sexual isolation is significantly higher than in comparable species pairs that live in different areas, allopatric pairs.
So the ones living together have stronger mating barriers.
Much stronger, especially when they are still genetically quite similar. It implies direct selection against hybridization is happening where they overlap.
Wow. And does this affect how fast speciation happens?
Dramatically. The data shows that sympatric populations can achieve full reproductive isolation at a much smaller genetic distance, meaning with fewer overall genetic changes compared to allopatric populations.
Let's pause on that. So sympatric species, maybe because of the selection against hybrids, can become reproductively isolated much faster.
That's the implication. The difference in the estimated time is huge. For these Drosophila, it might take only about 200,000 years for reproductive isolation to evolve in sympatry, driven by this reinforcement. Compare that to maybe 2.7 million years for similar levels of isolation to evolve just through gradual divergence in allopatry without that direct selection pressure against hybrids.
13 times faster. That's incredible. Selection against bad hybrids is a powerful accelerator.
It really seems to be. And there's experimental evidence, too. They took mixed populations of Drosophila pseudoobscura and D. persimilis, species that produce inferior hybrids. The researchers actively remove the hybrid offspring each generation.
Simulating natural selection against hybrids.
Exactly. And over just 5 generations, the frequency of females mating with the wrong species dropped from around 50% down to only 5%.
Wow, rapid evolution of mate choice right there in the lab.
Shows just how quickly selection can strengthen those prezygotic barriers when hybrids are costly.
Okay, so unfit hybrids drive faster isolation. But what if hybrids aren't unfit? What if, under certain conditions, they're actually better?
Now that completely changes the game.
Yeah.
Hybridization isn't always a dead end. Sometimes, especially in plants, it can be a source of new species, often through polyploidy.
Polyploidy, that's changes in chromosome number, right?
Yes, often a doubling or more of the entire chromosome set. It can happen when hybridization messes up cell division.
Okay.
And it's surprisingly common in plants. Estimates are like 40 to 70% of all plant species might have a polyploid origin.
That many. Wow.
The big consequence is that a new polyploid individual formed from a hybrid might be instantly reproductively isolated from its original diploid parent species, but fertile with other polyploids like itself.
So a new species, bam, in potentially one generation.
Effectively, yes, an instant postzygotic barrier. Evolution creating a new lineage in a single step. We see examples like Helianthus sunflowers in the U.S. A hybrid species, H. anomalous, seems to have formed from 2 parent species in probably less than 60 generations. It shows rapid sorting and stabilization of a new adaptive gene combination from the hybrid mix.
Fascinating. Let's bring it back to the finches, though, for an animal example. G. fortis and G. scandens on Daphne major. You mentioned song usually keeps them apart.
Right, song is learned, and it's a key behavioral isolation mechanism for them. Normally, hybridization is really rare, less than 1%.
But something changed.
Yes. There was an exceptional year with extremely heavy rains. This totally changed the vegetation and the types of seeds available. Suddenly, there was an abundance of small, soft seeds.
And this favored the hybrids.
Exactly. The hybrids, which tend to have intermediate beak sizes between the large-beaked fortis and the pointy-beaked scandens, were suddenly perfectly equipped for these new seeds. They had the highest survival rates, the best breeding success that year.
So the environment flipped the script on hybrid fitness.
Completely. And crucially, these successful hybrids didn't just form their own group. They started back-crossing with the parental species. Which parent they crossed with depended on the song they had learned, usually from their father.
Ah, so genes started flowing between the species.
Yes, that's introgressive hybridization. Gene flow across species lines, driven by the temporary advantage of the hybrids and their learned mating preferences. This event actually led to the rapid formation of what looks like a new, distinct lineage on the island, the G. fortiscandens group. It shows how hybridization, especially when the environment changes rapidly, can actually fuel adaptive radiation, not just prevent it.
Okay, so let's try and wrap this up. Pull the main threads together. Speciation is fundamentally about achieving reproductive isolation. That's the bottom line.
Absolutely, it's the defining feature.
And it usually gets started by geographical separation, allopatry or parapatric, that stops the gene flow.
Right, that initial split is key.
And then often, if hybrids between the diverging groups are unfit, natural selection can step in and reinforce the separation, strengthening mating barriers, sometimes really quickly.
That reinforcement mechanism, driven by hybrid inferiority, seems very important for speeding things up, especially where populations overlap. But we also have to remember the flip side. Hybridization itself, even if rare, can be a creative force. It happens in maybe 10% of animals, 25% of plants. It can introduce new genetic variation, create novel combinations, and sometimes, as with the finches or polyploid plants, lead to rapid formation of new lineages, especially when the environment is in flux.
It's like a genetic shortcut sometimes.
It can be. It adds another layer of complexity and opportunity to the whole process.
So the knowledge nuggets are clear. Reproductive isolation is the endpoint. How you get there can vary: slow divergence apart, or faster reinforcement when together, or even via hybridization sometimes. Which leads to a final thought, building on those timescales you mentioned. If sympatric speciation, driven by selection, can happen maybe 13 times faster, like 200,000 years, versus maybe 2.7 million for allopatric drift in those flies, it really makes you wonder, doesn't it? With humans changing habitats so drastically, moving species around, altering the climate, how might we be impacting the speed and the very dynamics of speciation happening right now all around us? Are we accelerating some paths, closing off others?
That's a profound question. We're definitely changing the selective landscapes and the opportunities for both isolation and contact.
Something to definitely think about. Thanks for joining us for this deep dive.